Hello, my name is Hairi, ID number 19432. I will explain about the introduction and equipment used for this experiment. So what is evaporation? Evaporation is a process where vapor is removed from boiling point to get more concentrated solution using evaporator. Evaporator is an equipment used to turn liquid into gaseous form. Liquid is evaporated, vaporized into a gas where more concentrated solution is remained. There are many types of evaporator equipment in industry, but our main focus is about climbing film evaporator, also known as CFE. CFE provides a high heat transfer coefficient and a short residence time for the solution being evaporated. Multitude CFE are used to concentrate solution that can be damaged from prolonged heat. Usually CFE will operate under vacuum to increase the heat flood and reduce evaporation temperature, and can be operated either in batch or continuous evaporator. Mostly CFE is used in food industry, such as to produce sugar, milk, orange juice, salt, and many more. There are several types of equipment involved in this experiment. The first one is feed tank. It is used to store water, recycle solution, and feed. It connected to the feed pump. Feed pump is used to deliver the feed into and out from the feed tank. Next one is climbing film evaporator, where it is used to remove the vapor from boiling point solution to get more concentrated solution. Cyclone is used to separate vapor and liquid, where vapor passing through a water-cooled condenser and into a receiver, while the liquid passes down into the concentrate receiver. Condenser is used to condense vapor into liquid state. The last one is transfer pump, where it is used to transfer concentrate to V12.
Hello, my name is Anita. I'm number 19039, and today I'm going to talk to you about the objectives as well as the results of the experiment.
The main objectives of this experiment is to determine the concentration of product concentrate of a one-stroke experiment of grape juice simulated with potassium permanganate and water mixture, and to perform a material balance on water mixture and calculating the thermal efficiency of the evaporator by collecting the product condensate and steam condensate during the experiment. To achieve the objectives of this experiment, we conducted the experiment by manipulating two variables stated earlier respectively at one time, which is feed level and pressure. Data are collected and graphs are plotted to enable us to see the effects of each variable clearly. While using the feed level as the manipulated variable, we have selected 4 different levels of potassium permanganate inside the tube, which are 25 cm, 50 cm, 75 cm, and 100 cm, with constant value of pressure in atmospheric. The first two graphs shows the effects of feed level on the evaporation rate by looking at the time for the first bubble to appear and for boiling to occur, as well as the concentration of potassium permanganate in the evaporate, concentrate, and condensate respectively. Similarly, the experiment is repeated using different value of pressure, which varies from atmospheric pressure to 600 mm mercury with increment of 200 mm mercury each, keeping the feed level of potassium permanganate constant. The graph of time versus pressure and concentration of potassium permanganate are as follows.
These are the results obtained from the experiment that is being held using the climbing film evaporator unit.
Hello, I'm Afiq, student ID 18810. Before we discuss about our results, let me explain how did we obtain those 6 graphs that we have shown to you just now. For the first graph, which is time versus pressure, there are 2 times that we need to record. The first one is time for first bubble to form, which means the duration from the beginning of the experiment to the first bubble formed. The second one is time to boil, which means the duration from the first bubble form to many bubbles form. After we have recorded these, we can obtain this graph. The second graph is concentration versus pressure. There are 3 steps to obtain this graph. First, we collect samples of concentrate, condensate, and evaporate. Then, by using refractometer, which is a device to measure the refractive index, we can get the refractive index value for each sample. We can convert the refractive index value to concentration value by using this calibration curve. It is a linear graph with the formula of y equal to 20.837x minus 27.802. The last graph is the volume versus pressure. This graph is simple to obtain. The volume of concentrate, condensate and evaporate are measured by using measuring cylinder. Repeat these steps to obtain the other three graphs for effect of evaporator feed level. So, that is how we obtain the results. Now let's move on to the discussion part.
Good morning, my name is Binita Chandrasekharan, ID 18802. So for today, I'm going to explain the discussion on the clamping film evaporator experiment. So, the discussion for this experiment is divided into three parts. The first part is the effect of feed level on potassium manganate solution. The second part is the effect of pressure on potassium manganate solution. And the third part is the concentration of evaporate, condensate and concentrate. For the first part of the experiment, the effect of the feed level on potassium manganate solution is studied by knowing the boiling time of the solution. So, when the feed level is increased from 25 cm to 100 cm, the general trend of the result is shown that the boiling point will reduce when the feed level increases. Furthermore, the time for first bubble to appear in the evaporator column will be shorter when the feed level increases too. By taking the feed level of 25 cm and 100 cm as reference, the boiling time is longer for the feed level of 25 cm, which are 194 seconds as compared to 191 seconds in 100 cm of feed level. This might be due to the reason that when the level of potassium manganate solution increases, the surface contact area of the steams and the solution is larger. Then, a shorter time will be taken for the evaporation process to take place. So, for the second part of this climbing film evaporator, the effect of the evaporator pressure to the first bubble time and boiling time is understood. In theoretical part, when the pressure of liquid decreases, the boiling point for the liquid will reduce, and indirectly this will shorten the boiling point. This is from Gionco Polish 2003. When the pressure are decreased from negative 200 mmHg to negative 600 mmHg, the boiling time for the potassium manganate solution will decrease. This is definitely caused by a higher negative pressure value that can reduce the boiling point of the solution. On the other hand, the time for the first bubble to form is shorter for the one with higher negative pressure, which is negative 600 mmHg. Based on the results, the concentrations of evaporates are always higher as compared to the others. Besides, by taking the volume of evaporate from the evaporator as a benchmark, the volume of evaporate will increase when the negative pressure value is lower and the feed level is higher. Thus, it can be explained that when the negative pressure value increases, the concentration of evaporate increases too. Conclusively, the effect of the operating conditions, which are the feed level and the evaporator pressure, are observed in this experiment, and they have shown a positive result for our experiment.
My name is Izian, ID 19343. Now, I will explain about the error analysis and conclude our presentation. The first one is parallax error, which is the feed level is not leveled correctly. To solve this problem, one should make sure their eye is perpendicular to the level scale to ensure accuracy. The next one is the human error, which is the spilling of evaporate, concentrate and condensate sample. To avoid spillage, one should handle the cylinder carefully and open the valve slowly. Other than that is the error in measuring the refractive index of the samples. The refractive index is not measured correctly as it doesn't fit the calibration curve equation. Before taking the refractive index, the sample should be cooled down first and make sure the refractometer is clean. Another error observed in this experiment is the feed is pushed up to the top column when we want to collect the evaporate sample. This is because the liquid is pushed by the pressure from the outside while the evaporator is in vacuum pressure. To avoid the liquid from entering to the top column, make sure that the evaporator is in atmospheric pressure by bleeding off the vacuum pressure before sample is collected. Last but not least, the recorded time was not reliable because it doesn't show a good curve. To improve this, make sure to immediately start the timer after the mainstream valve is open and push valve is closed, and note the time for the first bubble formation and when it starts boil. In conclusion, climbing film evaporator is used to study the evaporation of heat-sensitive products such as milk, fruit juices, and aqueous sugar. Climbing film evaporator provides a shorter time for the feed to evaporate and high heat transfer coefficient. Based on the results obtained, climbing film evaporator is best to operate when the feed level is high and when the evaporator pressure is low to obtain the highest concentration of condensate and concentrate. Last but not least, modification that stated in the error analysis must be done to improve the results of the experiment. That is all from us, Group 12, for climbing film evaporator experiment. We hope that you have gained some knowledge from our video. Thank you.